Thank you very much, Melody. I'm delighted to be here. Thank you to the organizers. The challenge is quite simple. Recent and future development in medical genetics. So easy, easy. I translated a little bit to the molecular basis of uncertainty because I really don't know what will be the future development of medical genetics, but also in the fact and the position that uncertainty is the daily life of geneticists, and you very know well we are dealing with risk, and risk is uncertainty. So what I would like is to overview, not delaying the dinner, but to overview some position of the complexity in medical genetics and some aspect that we would suspect might try to help solving part of the complexity presently and in the coming future. The temptation in genetics is simplicity. Monozygotic twins are very similar. When you breed stripes with lines, you get a crossed child. There is a mutant in the genome, so you have a disease. And when you have a disease, there is this mutant in the genome. In segregates with Mendel laws. This is very simple. There is also, for psychiatrists, I think it will speak by itself, and some authority in genetics. So it's written. There is a cipher, there's a code. It can predict. It's inherited, so it's addressing the past and the future. And again, it's written some sort of Pierre de Rosette. When you have the code, a genetic code, forgetting that it's not only enlightening 2% of the genome, but maybe less than that. But the general idea, the perception of the genetics, that it's written, so it's true. It's also cost-effective, and I won't expand on the fact that next-generation sequencing bring to a very low expenses the full genome. It's 1,200 euros, for instance, in Imagine Today. And there came a promise of genomic medicine by outstanding people. This is not ironic in my slide. I think these people will get the Nobel Prize for genome sequencing. But it was a bit of overstating that the building here with Human Genome Project, genome biology, genomics to health, to society, etc., and etc. are the skyscrapers here, was put forward in 2003. And our common understanding in medical genetics today, which is not a lot of people but worldwide agreeing, that it's a bit of an overstatement today. It was so important that in Montreal in 2011, it was the International Society of Human Genetics, one statement was put forward to discussion to the general audience. Personal genomes will become incorporated in standard care for all of medicine. Therefore, medical genetics will disappear as a separate specialty. I'm not conducting a lobby there to say we are alive, which still, that's not the point. The point is a certain amount of people worldwide do agree that they should not be rid of so rapidly. And I think all the talks, all the speakers today and probably tomorrow will agree on that, because we need some of those people still yet dealing with AI, fine, dealing with computer-assisted science, fine, but to address the patient with this information, I think it's so important. The vast omission of these statements was complexity. Thank you, Thomas, for this slide. It's also true for any single-cell genomic experiment. It's complex. It's not so easy. It's not simple. And the more NGS data you have, the vaster is complexity. Complexity is in variability. That's the vision we have. I mean, natural vision where there is a mutant in this patient. The truth is there are a whole bunch of variants on varioning this mutant. And what does tell you that this variation is the one you are looking for, and what does tell you that the other variations next door would not play a role in the phenotype of the patient. The complexity results in a very variable human genome landscape, and this is very interesting. There is put forward some science in population genetics and genetics migration. You can use this variation, both single nucleotide variation but also copy number variation. It has been the topics of today. I will not expand on that, but not only for disease but also for population genetics and many other aspects, very useful. But still, it remains that it's very difficult to find a way in such a genome where a gene is actually in a desert, surrounded by variability of single nucleotide sequences, but also copy number variations. These variations are submitted to selections, and again it helps us to retrace the history of our population and migration and the drift of population with neutral variants. But what can tell you that a variance is a balanced variance with a deleterious effect or an advantageous variance that has helped our population or some of our population to adapt during evolution across the centuries. Another point about the complexity is the low number of genes. 20,000 in human, Drosophila 13,000, C. elegans 18,000. And Arabidopsis thalaina, a little plant of the landscape, 25,000. Let's remind that banana has 69 chromosomes as well, so it's quite humiliating, but it's the truth. So in a way, the big is not always beautiful, and I think this is a very important comparison here in 3D graph with genome size in the x-axis, in the x les abscisses quoi, or the y-axis with the coding versus non-coding base pair, and the big fact about evolution, if the complexity of the species is presented here, one cell, few cells, multicellular, eukaryotes, and human, the major effect of evolution is the increase of non-coding DNA sequence. And basically, it was human genome is so complex, and maybe why the biology of human is so complex and important. So complexity is not only gene number, which drives us to think that when you are analyzing proudly an exome of a patient, it's 1.8% of the genome, that 8% of the genome is under selective pressure for selection with probably functional aspects, and so importance. And that if you look at the ENCODE consortium, 38% of the whole genome, which means 80% of the single copy genome, it has some features of function, whether biological or evolutionary, indicated by conservation of the sequence. So it's a lesson of humility for us geneticists. We have to explore much broader than the coding sequence and into the non-coding DNA. A third aspect, and it's been approached today very much, I wouldn't say plasticity, but rearrangement of the genome by de novo mutation, and any child from any healthy parents has a mean of 70 to 80 novel single nucleotide variation, one de novo mutation per exome. Does not mean it's deleterious. We are all survivors, by the way, and one CNV for the child. So de novo mutation is the real life, and it's difficult to explain to patients, and counselors here know that, to explain to patients that they have not been targeted by a taboo or something which is a disaster, but they have been the unlucky results of a rain of mutation that everybody gets in the duration of the conception of the embryo. And it's no surprise for the audience here that these de novo mutations were so important in deciphering the molecular basis of some autistic spectrum disorders. Another line and dimension of complexity is epigenetic. The genome is not, I told you, not an alphabet, but it also has to be expressed. And what regulates the expression of the genome by non-coded primary DNA sequence changes, which is reversible, which might submitted to somatic transmission and sometimes from transgenerational effects. The epigenetic variation, both mostly based on DNA methylation and histone modification, came to the scene recently as possibly involved, probably involved, much more than we expected in the phenotypic variation in human, non-depending on the genotypic variation. So two textbook uncertainty case studies, the variable expression, age of onset, 56, 43, 44, 25. Why? So the variable expression expressed in measured by quantity as the age of onset is in the textbooks of genetics from years. Something, well, variable expression, but why? Penetrance, which is the probability of having a phenotype given that you have a genotype, so a very simple ratio, is represented here in a family with an autosomal dominant trait, and 3 individuals here do exhibit the genotype, but not the phenotype. Why? And this is very important not only for risk calculation, not only for the perception of the family of somebody affected and the other not affected carrying the same genotype, but also for treatment. If you understand why these individuals would not show the phenotype, you may have some tracks to have thoughts about what could reverse a phenotype. So one of the first answers was, and still is, complexity is probably the allelic and loci heterogeneity. It was one of my heroes in genetics. I think Thomas is also one of your heroes. John Burden Sanderson Haldane, one of the rare communist British gentlemen, who made also a very great discovery about diving under the water at 8 meters with a submarine, and he got deaf by explosion of the tampon. Anyway, it was an outstanding geneticist, and he referred with a very old drawing, which is his drawing, about this complexity being the result of one genes, different mutation, or different genes for the same phenotype. Yes, that's true, loci heterogeneity, mutation effect, gain of function, loss of function, and the allelic combination at a single locus in autosomal recessive inheritance are clearly part of the answer to complexity. But it's clearly not the single answer. So I want to survey some of the few examples, sorry for you because it's very textbook driven, examples of what we can imagine or know from time to time that would add to the complexity independent of the allelic series and the locus heterogeneity. Usual suspects. The first one is allelic expression. I think this is one overlooked way of the expression of the genome by transcription as compared to the pure sequence. At a disease locus, when intrafamilial variability is inferior to interfamilial variability, you might expect that the dosage of the alleles could play a role. This is known for I try to pick up some examples from neurogenetics, but not always. For spinal muscular atrophy, where the dosage of a pseudogene might compensate partly or completely the full-blown deletion of biallelic SMN1 copies in patients. So this patient is unaffected with 4 SMN2 copies, the pseudogene, compensating the full deletion of the 2 alleles. And this looks like the segregation as an autosomal dominant inheritance, which actually it's a recessive disorder, combined with a compensation at the same locus, but by a pseudogene. Again, this gave really interesting clues for therapeutic options, which are to reactivate the pseudogene, and there are clinical trials, you know that, trying to reactivate, to compensate SMN1 deletions by SMN2 overactivation. one allele or two, all these monoallelic expression variants with splicing variant, editing, promoter variants, targets of microRNAs, etc., could be some explanation to the variability at a single locus, independent of the disease-causing allele. Another suspect is a dominance effect, which means that if you are in a dominant pattern of inheritance, the trans allele may be of interest. We had this result in Hirschsprung disease. It's neurologic, but it's the enteric nervous system, second brain. Not interesting, but very complex. Much more complex than central nervous system, of course. Anyway, when you have a segregating a full loss of function mutation of the RET proto-oncogene in this family, these patients do harbor the phenotype only if the trans allele carries a predisposing allele, which gives this type of pattern we deciphered with Aravinda Chakravarti of families in between autosomal dominant with low penetrance and oligogenic inheritance, which actually segregates a disease-causing variant. It's a full-blown stop mutation here at RET that express only if it's combined with a predisposing allele in trans on the other allele contributed by the non-carrying parents. So this is an extremely long Hirschsprung disease with the variant plus the modifier in trans. I remember Stylianos Antonarakis trying to solve some of these examples of patients with 22q11.2 deletions and schizophrenia by attracting the non-deleted allele for targeted sequence, trying to find some, in those 3 megabases deletion, some predisposing factor to schizophrenia in those particular patients. And this is the paper by, a referring paper by the group of Husman. Somatic genetics. Not all alleles are expressed in all cells. And if they are by germline mutation, they can vary. This is known by reversion mosaics in some immunologic disorders, when the revertance in the cell can explain some variability in tissues over life, across life. So it's also important that the disease can vary during life because somatic mosaics can incorporate a reversion of the mutation. It's not very frequent. I'm not telling you about frequent examples, but that does exist and shows that it's not simple. The instability of repeat mosaics in myotonic dystrophy. And, of course, with X chromosomes, the inactivation and the lyonization mosaics of X chromosomes in Duchenne muscular dystrophy, very simple, with affected boy Becker, the dystrophin is present, is absent with Duchenne. What is that? It's the reversion of the mutation by deletion and recombination in some fibers. And the carrier females here, with 4 female carriers, and this female carrier is an extreme lyonized on the inactivation of the normal allele with a very close pattern of immunohistochemie of dystrophin in the muscle in the F panel, the F panel here, looking very much like a boy. And we have some example like that. This is a series of triplet, spontaneous pregnancies, 3 premature daughters, the diagnostic of Duchenne muscular dystrophy in a female here. Imagine how the neuropediatrician has to think about that diagnostic. And one of them is carrying the mutation. It's a mosaic in the parents. Genetic is simple. This lady is unaffected. These 2 are monozygote. And the lyonization made Duchenne muscular dystrophy in this girl and not her monozygotic twin sister. Complete mirror inactivation. This is 100% bias favorable to the girl. This is 100% bias unfavorable to the girl. So the mirror image of lyonization. My feeling and the feeling of those who reported this observation, Marlène Rio and colleagues and the colleagues in Cochin, is that it's not randomly. It's not a random observation. This mirror image of lyonization something has to do with twinning, but we don't know why. Transmission effects, you know very well the old stories of variegation. You could have the mosaic of gene expression depending on regulatory sequence rearrangement without alteration of the primary DNA sequence and via modification of the chromatin environment. Parental imprinting in Prader-Willi syndrome, in Angelman syndrome, the parent of effect will modify the phenotype in the child. We thought we had something like that in Hirschsprung disease. This is a support family picture of a meeting with a vast increase in males as compared to females. This rings bells in neuropsychiatrists as well, but it's also true for enteric nervous system. And we felt it could be a parental bias transmission of the predisposing RET mutation, and actually it's not imprinting, it's just a differential reproductive RET involved in RET transmission through meiosis, which will bias the transmission through the female lineage. So these patterns of gender bias are also very tricky to decipher. Interaction, and we're coming to multiple gene loci, interaction with unlinked loci, epistasis, gene-gene interaction. Of course, when you have unlinked modifiers, sorry for the Y, intrafamilial variability would equal interfamilial variability. And of course, again, Haldane predicted the relative importance of principal and modifying genes in determining some human diseases. It was in 41. Very few things have changed in this talk, I'm afraid. Plastine for spinal muscular atrophy, again, an example in neurogenetics. Those deleted females rescued by an abnormal expression of the plastine-3 gene on the X chromosome, which is associated with SMA, spinal muscular atrophy protection, in some discordant family for the phenotype, as a sex-limited protective modifier. Many, many other examples in cystic fibrosis, sickle cell, you name it. And so it's clear that the rest of the genome, unlinked to the disease locus, plays clearly a role. Again, the field conductor of Hirschsprung disease, we have this impact of RET variant predisposing to Hirschsprung disease and acting long distance on the expression of mutation at other loci, FOX2B for Oninsker's, which will present Hirschsprung disease or not, depending on the genotype on chromosome 10. The same thing for Down syndrome, trisomy 21, Down syndrome, exhibiting Hirschsprung disease or not, depending on the chromosome 10 genotype. And finally, gene-environment interaction. You have many examples in psychiatry. We'll just pick one, is prenatal exposure to valproate. And this is a slide from Dominique Stopaglione and Curie. I think this is incredible. This is incredible and terrible. These are the penetrance of BRCA1 mutation in the same families. So let me explain well that these are the mothers in the red curve, the blue curve, sorry, and the daughters in the red curves. So they have the same mutation, BRCA1 exposing mutation to breast cancer. And if these women are born before the 40s, the penetrance, which means the occurrence of a breast cancer, is that pattern. When they are born after the war, the penetrance is that pattern. And they are the ladies within the same families with the very single BRCA1 mutation. Smoking is a large effect. Parity has a large effect, contraception, but there is still a mystery about that, about what in nurture might modify the pattern of expression so significantly between women born before the war and after the war, exhibiting the same mutation that they transmitted to their daughters. I'm always very surprised and frightened, I might say, by this pattern of difference in penetrance in these pedigrees. I just came by car to the meeting and I got a call from my mother, just discharged from the hospital. She's very well. Very, very well. Anyway, and they called me and said, I'm out, I'm fine, I was Pitié-Salpêtrière, don't worry. There was this nurse, she was delightful. She was a beauty, a Venetian blonde, and she has all skills for care. Please, Stanislas, don't tell me that innate is equal to environment. My mother. So I told that I would come and tell you that story. What should we expect from that viewpoint? And I would try to tell you what is not expect by plea for, what I want to plea for. First, I think that Stylianos and Jack Beckman think that we should pay more attention still to Mendelian disorders. They are very important to address lessons that we can, in a second time, transpose to complex disorders. So I think we should do that. Second, modeling disease is extremely important. I will not plea for that in Sainte-Anne and the Centre of Neurogenetics in Sainte-Anne, of course, but to model disease in cell by reprogramming, differentiating, to reproduce neurodegenerative disorders, and why not psychiatric disorders in animals and cells is very important. Edor Kabashi was recently recruited in Imagine for the neurological disorder translational research using fish as a model. So it's fine. It could be Drosophila, it could be fish, could be mouse, but this circle from phenotype to trials pass by experiments in the models, and I think we should cherish the very classical models of Morgan and others a century ago. The third hint is that we should explore DNA beyond the primary DNA sequence. It's obvious, sorry for that, but expression of the genome, the chromatin marks, by whatever techniques the ENCODE consortium proposed, are very important. So we cannot approach a complex patients or a complex mode of inheritance with only the sequence. I think the chromatin marks, the RNA-seq, the attack-seq, the transcription of the genome are very important, and all geneticists came across examples where the finding the disease-causing anomaly came from expression studies and not sequencing studies in the first place. A few examples about that. We are studying the SOX9 locus in craniofacial anomaly, never mind, and found that the mutation of SOX9 in the coding sequence result in a very severe polymalformative syndrome, which is lethal, campomelic dysplasia. But when you have alteration 1 megabase of DNA upstream the SOX9 sequence, impacting an enhancer, you could have a single part of the syndrome. You could reduce the syndrome in a symptom. And this is true for SOX9, this is true for sonic hedgehog, for TBX5, for this gene in development. When mutation so distant to the promoter, 1 megabase, 1.4, 1.2 megabase from the promoter, are actually impacting the regulation of the gene in a targeted tissue-specific manner that would result in part of the syndrome and not the full-blown syndrome. I think this mechanism that we've proposed as enhancer modularity, it's a little arrogant, but I think could be expanded a little more to try to understand the part of the syndrome when you can try to decipher the syndrome in small pieces. It's true for enhancer alteration, but it's true for landscape of the genome alteration by 3D dimensional DNA analysis and chromatin marks, and for the same gene, SOX9. Stefan Mundlos in Berlin demonstrated that sex reversal could be ascribed to complete inversion of duplication of a sequence of DNA upstream SOX9, who's actually the primary DNA sequence is not changed. It's just duplicated or inverted, so you could not pick up by sequence. But if you look at chromatin marks, it displays the topologic associated domains of regulation and deprives SOX9 of its regulator or include regulators that should not be there. So this complexity of dysregulation of the genome by large domains impairment is, I think, a very open story, which is also supported by, of course, biology of these archipelagos of regulation in gene desert domain. And we should bear in mind that most of the genome-wide association studies hints are located in non-coding region of the genome. So it could well be that the GWAS at last are true, but with molecular basis that they indicated by an association marker, but the molecular mechanism is much more complex than single DNA, primary DNA sequence change. Of course, and I think it's hope Sebastian and Thomas share that, the annotation of the genomes and the ascription of variants to their role is so an important endeavor of genetics today. Variant in an exome, 1.8% of the genome, to 24,000 variants in an exome, one per gene, just a simple reminder. All these variants are loss of function by truncating mutation. So again, we are survivors. And these variants, most of them are singletons by the EXAC and GNOME-AD database. So it's not a surprise to find truncating mutation in a patient. It's an obligation. So which one is the disease-causing one? So that's a great point. And what Gary Cutting recently in the American Journal published is that the annotation of DNA variant is a major goal of human genetics, if not the major goal, because the cost of sequencing decreasing, the production of NGS sequence increasing, what's the gap which is increasing is not the annotation gap, but the interpretative gap. And this is very important to have in mind. So this drove many institutes, and we devised, not we, Patrick Nitschke and the Paris Descartes University, I imagine, devised a software suite to try to help the interpretation of genomicists and geneticists by polyexome, polydiac, déjà vu, many tools based on AI-supported annotation of panels of exomes and soon, hopefully, genomes. And even when you have an exome, this is Louis. Louis has intellectual deficiency, dysmorphic features. I'm not asked to face to gene yet. Anyway, no one of us could recognize a syndrome. And this is the result of Louis' exome. This is true. SYN1 gene, double variation. It's a missense, but very rare, probably impacting. The prediction softwares say it's disease-causing, it's impacting the gene. Fine. And it's a recessive pattern of inheritance. And the mother is, of course, pregnant. And, but Louis has also a de novo mutation of ROC1. ROC1 is not known for disease-causing gene, be a disease-causing gene yet, but when you look at the expression pattern of ROC1, at the function, presumed function of the gene, it could be interesting as well. And Louis has also a benign, so-called benign, X-linked variant of UBE2A on chromosome X, known to result in some intellectual deficiencies with syndromic features. And so what would you counsel to Louis' parents and this pregnant woman? Your turn. Okay, so engage in counseling. I was extremely happy that from Psychiatric Institute and Neurogenetic Institute you would promote this seminar because genetic testing is extremely difficult in the individual situation, in the context of the family, and also in a population. I think this is very important. We have to cherish population and epidemiology genetics, but maybe more to cherish population and epidemiology geneticists. They are very rare. There is not an extinct species, but they have been overwhelmed by data, by NGS sequences. And these people and the French school and British school and American school are very strong. And these people are not well listened to as far as population genetics, heritability, polygenic risk scores, Mendelian randomizations are concerned. I think we have to listen more to these people for their lessons and the precautions they take to say that a result in human genetics is true and could be carried to relative risk, to odds ratio, and to counsel families. Again, it's not ironic, it's not passéist, it's not the discourse of the past, but it's the present, the reality of the present. I would so much love that some AI device would tell us the risk scores of Louis in this observation. But the truth is that we don't have that yet. So please, we have to cherish those people and their instructions and teaching, not only for nature versus nurture, but also for the real design of the architecture of genetic disorders, which is really a challenge. Not to forget clinics. It seems obvious. The machine is fine, but the next generation sequencing demands next generation phenotyping. Thank you, Raoul and Leslie. I think everybody is now convinced that these results are very important to challenge with the clinics, if not the clinics here, but at the first place, at least to challenge the genetic results with the clinics. This is the warehouse we have devised in Imagine, with now more than 6 million documents, 679 patients included, 200 databases, and these, we discussed that with Thomas recently, and this is some low AI grade recovery system where you can design a cohort, annotate patients, to get similar patients, sometimes to get an undiagnosed patient to diagnosed patient, and also to merge the phenotype with the genotype, which is our challenge here. And Nicolas Garcelon is developing that program, Doctor Warehouse, of course, for rare disorders. And this is, I think, a very important initiative to combine those very basic data with very good, very solid clinical information. You know, the GIGO syndrome, garbage in, garbage out. So the clinical information is important. It has to be good. And these are the medical records of Necker Enfant Malade for 10 years. I think the interns, the residents, the clinical assistant, professors are very good. And this is the quality of the Medicare in Europe and North America. So please, we have to use the quality of the excellence of the phenotyping in this type of database. This is a study Arnold has carried out for 20 years in the what he called the mobile regional genetic clinics, CMRG, with the help of the Agence de Santé Île-de-France, with Necker, L'élan retrouvé, which is a consortium of institutions for autistic patients, trying to progress with the clinics, recognizing some syndromes, Kabuki syndrome here, Smith-Magenis, with the genetics and also with brain imaging. And in a nutshell, 20 years, 451 patients, he's seen almost himself, 26 institutions, on-site consultation to say, well, we will not extract the patient to a very strange, unexpected environment, but you will do it on-site. And less than, the conclusions in a nutshell, less than 1% declined the offer. Brain MRI showed up to 25% anomalies in those in whom it could be carried on, 251 in autistic spectrum patients, which means that the fact of being on site and to have the parents in the decision making for MRI or not was very important. Previously undiagnosed genetic condition recognized in 75 of them, CNV variant, Fragile X, disease causing, that's not very important. And all these patients has a trend, this is very well known, to have moderate to severe ID, the one with the diagnostic. We know that, but I think this initiative from Arnold was interesting to say, well, we have to carry at the same time a very good clinical ascertainment, the brain imaging and the NGS testing. Another scope, and maybe I will finish by that, is the therapeutic option. Genetics was contemplative, I would say. It's now acting, it's not as a sole specialty with the other specialties, of course. Just 3 flashes and vignettes on repositioning, cell therapy and gene therapy. Repositioning, this is Laurence Legemallet in Imagine. She has devised the analog of effect of CNP on achondroplasia, vehicle, CNP, and the increase in the bone length. So there is a phase 2 to 3 trial now in Western Europe ongoing based on repositioning a molecule to make the achondroplastic patient grow. Alain Ovnanian, transplantation of skin by gene therapy modified in epidermolysis bullosa, again a phase 1 trial with COL7A1 locus, and very recently Première mondiale by Marina Cavazona on gene therapy on sickle cell. It's not to claim that by arrogance, we can do it, but it's possible. It's not impossible. That's just the take-home mention. It's not arrogant, just it could be possible. So I think it's the seventh pillar for future of genetics. We have to be treatment-orientated. The last but important question is what could be the model for an institute to devise this system, not to be the best, but to try not to forget something in the ecosystem that would make you pass by a good idea. The genocentric model is fine, it's genetics as a unique platform in the Ile de la Cité in Paris for everybody. The clinical sufficient or arrogant model is that we have the platform in Évry, but we geneticists are clinicians in Paris. What we try to devise in Imagine, and not in Imagine only, I think it's many other institutions, is to have clinics and genetics in the same place, in a unique location, where we can have the clinics of the patient, this is Imagine itself, the physicians, the referral center for rare disease, the core facilities, the research labs, and the support teams. It could not be done everywhere, but we think that to gather in single place can accelerate things. It does not make people genius, but it makes people just to speak to each other at the same place. This is what you do in Sainte-Anne with the Neuroscience Center. This is what we try to do for rare genetic disorders in a pediatric setting in Imagine, to accelerate this virtuous loop from patient to basic research and back to the patient. This is my conclusion slide. What we plea for is those 7 pillars, but also to have a close relationship between basic and translational science, research and clinics, good practice and not practive in genetics. I think it's important. There are so many fake news in genetics that this merging of scientists and clinicians is also some resistance to fake news, and also to try to merge public and private funding. It's an evidence. And to finish, because you asked me about the future of genetics, 2 sentences and quotes about the future. It's difficult to make predictions, especially about the future. And from the Secretary of State, there are no knowns. These are things we know. Follow up, it's very difficult. Secretary of State. We also know there are known unknowns. That is to say, we know there are some things we do not know. But there are also unknowns. That's one we don't know. And I think there are many things in genetics we don't know. Thank you for your attention.